Three major parts were identified in the ear. Part 1, external ear or outer ear, made up of an ear lobe. This is also called auricle. Meant for directing sounds into the meatus. Meatus is a canal. We have referred to it, and we also said that the meatus is protective. Not only it conducts sound waves, airborne sound, and we said it has ceruminous glands which secrete wax, waxy cerumen, which accumulates, dries up and falls off on its own in a majority. In a minority, it doesn't get dried, it accumulates there due to loss and may cause loss of hearing. Also, hairs there. Well, in general, the meatus is meant for protecting. End of the meatus is marked by a tympanum. Tympanum is eardrum, a tightly stretched membrane, very thin membrane, across the external auditory meatus, wherein you find can receive vibrations, airborne vibrations. Externally, that is on the meatus side, it is covered by skin, and on the inner side, it is covered by an epithelium. Since it can vibrate, it can convey the vibrations of sound to the next part of the ear, which is middle ear. The middle ear is a cavity which has three components inside it. A bone shaped like a hammer, another bone shaped like an anvil, and a third bone shaped like a stirrup. Malleus, incus, stapes, MIS. These are the smallest bones in the body, are identified as ear ossicles. Ear ossicles are movably articulated with the help of small muscles. In fact, the malleus is attached to the tympanum, the incus to the malleus, and the stapes to the malleus. Stapes has two attachments. One is to the incus, and the other side of it you find it is attached to the part which is called oval window. Oval window. Lower down you find there is another which is called round window. Let us see to which this is attached. So, three components are there. Very funny. The ear has three components. The middle ear has three components. The inner ear has three components. Looks as if we have components of multiples of three. The components of the inner ear inner ear, internal ear are involved in two functions. I pointed out how the whole inner ear is a membranous sac, that which can be distinguished into three different parts. And the whole bony area enclosing the membranous, tubular, vesicular components, these are floating in the fluid. The fluid in which these components are floating is the perilymph. Perilymph. The tubular and the vesicular components also contain a fluid inside them, that is endolymph. Perilymph and endolymph are just the two terms and not to be confused with the lymph of the circulatory system. Understood what I said? The word lymph here used as a suffix should not be confused with the lymph of the circulatory system. This is something different. The compositions of the two are different. Perilymph and endolymph. So, the whole bony capsule located just behind the eyeball, the temporal bone of the skull, has the same shape as the tubular and vesicular components of the internal ear. That is why it becomes extremely necessary not only to identify and to clearly distinguish the two components. One component is the bony labyrinth. Labyrinth is a maze. The other is the membranous labyrinth. Bony or osseous labyrinth. The other is the membranous labyrinth. The bony labyrinth takes on the shape of the membranous labyrinth which it encloses. The fluids are necessary. You know, wherever the fluids are there, at joints here and there, everywhere it is protective and lubricatory. Here they have an additional function. Additional function is to convey sound waves. So the sound has to pass through several media here, isn't it? Yes. Airborne sound touches the tympanum. From the tympanum onwards it is passed through solid bones. From the solid bones it is passed into the perilymph. From a perilymph into the endolymph. Endolymph, let us see what it is. So, external ear, middle ear. The problem with the middle ear is that it is connected to the throat by a pharyngotympanic membrane, formerly called eustachian cavity, eustachian tube, more or less horizontal in young children, and in adults find it is slowly inclined. It is generally kept closed and open when we yawn or when we open our mouths to eat. Now this helps to equalize the pressure within the middle ear and the external ear on either side of the tympanum. The pressure equalization is very important for the functioning of the tympanum, for its vibration. Any disturbance in this would mean pain in the ear, as I said, which some of us experience in air travel. I shall draw the third component, inner ear, internal ear, bony labyrinth, Inside that, a membranous labyrinth. I had just drawn. Looks like a rough figure. All right, let me repair it. Once again, we are burdened by the number 3. 3 components once again. 1 is called vestibule. 2 semicircular canals. Cochlea. Vestibule, semicircular canals, cochlea, all of the bony labyrinth. Oh. Each containing a duct system. For example, the vestibule is made up of 2 sacs, membranous sacs. One is called the utriculus, other is called the sacculus. Utriculus and sacculus. Semicircular canals contain 3, once again 3. Semicircular ducts. The cochlea contains the cochlear duct. The utriculus, sacculus and cochlear duct are components of the membranous labyrinth. In this, where do the semicircular canals? Well, though we have written 3 distinct parts, they are all interconnected. One, two, filled with endolymph. Interconnected and filled with endolymph and contain the sensory organs. Utriculus and sacculus in this contain sensory organs involved in equilibrium. Semicircular canals, utriculus, sacculus contain sensory organs associated with equilibrium and acceleration, turning of the head movements, static equilibrium, dynamic equilibrium. In general, those parts generally called the maculae are associated with equilibrium. The cochlear duct has sense organs associated with hearing. Three parts: vestibule, semicircular canals, cochlea. In this, cochlea is the one that gets its name from a coiled shell. Here, the coils take about 2.5 turns around a central axis. 2.5 turns around the central axis. You cannot make out the structure of the cochlea if it is coiled like that. What should we do? We must draw how it is straightened in its straight form and in a TS. Two drawings would be required to understand the structure of the cochlea and the sense of hearing. Two more drawings would be required in order to identify the semicircular canals and the maculae of the utriculus and sacculus. Maculae of the utriculus and sacculus. First, let us understand what the semicircular canals are and what semicircular ducts are. Semicircular canals belong to the bony labyrinth. Semicircular ducts belong to the membranous labyrinth. Semicircular ducts contain the endolymph, semicircular canals contain the perilymph. Three canals are there. One anterior, posterior, back portion, lateral. So one is like that, one is like this, and one is like this. Three semicircular, very small, of the size of a split pea. Split pea, of the size of a split pea in humans. Semicircular canals have ends wherein you find they are enlarged. The enlarged end of each is called an ampulla. This is the place where what is found? Macula. Macula is the sense organ associated with equilibrium and acceleration. So here, and then that is the sacculus, then turns. Semicircular canals then and their semicircular ducts contained within them contain the maculae which are associated with the sense of equilibrium. How about the cochlea? Cochlea is coiled, isn't it? This is the round window, oval window. Shall we straighten this up? Unless we straighten it up, as I said earlier, unless we take a TS of that, straight tube. This contains perilymph. This is just a tube, cochlear tube. Containing endolymph. The two cavities, one situated above and one situated below, scala. This is scala vestibuli. With the right perilymph. This is scala tympani with perilymph. This is a tube, cochlear tube, scala media, the median cavity. So this is continuous. Scala vestibuli, scala tympani and scala media. Scala media, middle cavity. Tympani and vestibuli are continuous at the end of the cochlea. It's called heliotroma. This area is called heliotroma. This is the one which contains endolymph. Now, how do you understand this? Unless we take a TS. TS is absolutely necessary for two reasons. One is that it tells us where the hearing apparatus is located. Second is that the three divisions of the spatial organization of the two is visible only in a TS. So shall I take a TS somewhere here? Just see. Please remember the figure I have drawn earlier. I shall redraw it, make it bigger. That is the os boni labyrinth. 1, 2. This is scala. This vestibuli. This is scala tympani. This is scala media. This is the vestibular membrane. Also called Reissner's membrane. This is another membrane, tympanic membrane, tympani, vestibular membrane, tympanic. This is to be not to be confused with the tympanum which I referred to, scala media, and then This is the tectorial membrane. And this is the organ of Corti. This contains perilymph, this contains perilymph, this contains endolymph. So actually this part of it is this is cochlear duct. The rest of it is a bony labyrinth. This part of it is cochlear duct, rest of it is bony labyrinth. Whatever contains endolymph is a part of the membranous labyrinth. Whatever contains perilymph is a part of the bony labyrinth. Let us make it very clear. There should not be any confusion. So, instead of now referring to various other parts, I shall retain only the part involved in hearing. Not that the fluids are unimportant. Well, here is tectorial membrane, and this is the organ of Corti. We have almost covered the structure. It is necessary to go back to the two windows. The organ of Corti is located on, this is endolymph, remember, and there is another tectorial membrane over that. It's present throughout. What was not mentioned here is a part of the nerve. A nerve involved in hearing is auditory nerve. This is the eighth nerve, purely sensory. Eighth, cranial nerve. So fluids are there. Fluids respond to sound waves. Let us draw this here. Why do you bend it? I am bending it. The bending caused by a depression produced by a sound wave. The wave, entire membrane responds. The whole tube responds to a sound wave arriving at the oval window. Through the agency of the stir. The fluids also vibrate in response to the sound. They create a wave, and the wave directly affects the membranous labyrinth containing the middle part, which is called the scala media. Scala media has the organ of Corti. Organ of Corti is a complicated structure made up of, shall I show here? I will draw the cell separately. Columnar cells with butting out a specific term not used anywhere in biology. It's called a stereocilia, and the expression hairs are written in inverted commas. These are not hairs because hairs themselves are multicellular structures. Stereocilia, we have heard about cilia, we have heard about microvilli, internally supported by microtubules. Stereocilia are of the same type. They have a structure very similar to the cilium or flagellum, and an important part is that the stereocilia butt against the tectorial membrane. Practically in contact with it, these are stereocilia, so tectorial membrane. Any change in the orientation of the tectorial membrane, I repeat, any change in the orientation of the tectorial membrane is immediately felt by the cilia and conveyed to the cells, and you find what starts there are the sensory neurons. Sensory neurons which combine together to form the auditory nerve. You can guess. If sensory neurons are there, there must be an action potential. The action potential must pass from there and then go into the peripheral nervous system. The peripheral nervous system must convey the message to the brain proper, which enables us to hear. We are referring to the hearing apparatus. The magnitude of sound, as you know, that can be heard varies from one person to another. It is generally mentioned in decibels or hertz, Hz. You know it better than I do because you study a whole chapter on sound in physics. Anyway, what should be the sequence of hearing, if you ask? Well, sound waves, airborne sound waves strike the tympanum. From the tympanum, the vibrations are conveyed through the three auditory ossicles to the oval window. From the oval window, the vibrations are conveyed into the perilymph. The perilymph responds to the sound by movement. Can't be compressed. No fluid can be compressed. So, the whole sound wave travels from one space, scala to scala, scala to scala, through the heliotroma end. And then during the passage of this fluid, which has created waves, something like the waves which you see in the sea water, you find the whole tube, what tube? Cochlear tube, responds to it, and the response is picked up by the organ of Corti, to which the tectorial membrane responds, and the whole response is conveyed by neurons, sensory neurons, to the peripheral nervous system, 8th auditory nerve. From the auditory nerve it reaches the brain. Well, that is how the hearing is explained in a simple manner. Any one of these can become an item for a multiple choice question or a assertion reason question or a column 1, column 2 comparison question. For example, the simplest, the cochlear duct contains number 1, endolymph, number 2, perilymph, number 3, organ of Corti, 4, 1 and 4. So, this would be the correct answer for a question that is framed like this. Organ of Corti, you can have a question. The cells of organ of Corti bear cilia, stereocilia, flagella, tympanic membrane. Assertion. Okay. The ear ossicles in the middle ear occur in the sequence incus, malleus, stapes. Reason are the stapes is attached to the round window of the inner ear. How are these two statements? Please. The ear ossicles in the middle ear occur in the sequence incus, malleus and the first is not incus, first is malleus, then is incus, the third is stapes. So this statement is wrong. The stapes is attached to the round window of the inner ear, stapes is attached to the oval window. So this is also wrong. So options are there, both A and B are correct. Both A and R are correct, and R is the correct explanation for A. Number 2, A is wrong and R is correct. Number 3, A is correct and R is wrong. Last one, D1 is that both A and R are wrong. So this is one way of coming to grips with some of the facts of the ear. You can have a column matching with any one of these components mentioned there. For example, perilymph has a higher concentration of potassium ions than the ICF, or it is similar to the ICF almost. And then you can give any one of the 3 ear ossicles. You can also refer to the pharyngotympanic membrane, or you can refer to the auditory nerve innervating the organ of Corti. So a number of different objective questions can be made out of this. The second function which the internal ear is carrying out is balancing or maintaining equilibrium. Maintaining equilibrium is a function of maculae. Maculae are sense organs capable of detecting pressure, altered pressure. Maculae. So there are maculae in the ampullae of semicircular canals. I'll write ducts. And in the utriculus sacculus. The differential impingement created by changes in position of the body head. Head is particularly important as it rotational movements and nodding movements. These are all detected by the maculae located in 2 places, ampullae of the semicircular canals and the utriculus and sacculus of the vestibule of the internal ear. Though you find the organs of hearing and the organs of equilibrium are continuous with each other, the functions that they perform is going to be different. With this, we end neural integration and coordination, and we pass on to chemical integration and coordination. Chemical integration and coordination. Chemical here refers to hormones. We are very often excited by a movie of Schwarzenegger or of some other person. There is a series of action-packed movie. I am asking you, why should you go in for an action-packed movie there on the TV screen when your own body is making such an action-packed movie every minute and now? It's happening. Our biochemical reactions are nothing less than the action-packed movie of some film star. Take, for instance, an insulin molecule released by the pancreas. It travels in blood, reaches the target cells, binds to the surface receptors of the target cells, enters the cell, and then brings about changes in the metabolism of the whole cell. Is it less exciting than the one thing that you see there? So our body itself is a bundle of chemical reactions. It's a chemistry laboratory where you find every moment exciting things are happening inside the cell. Now, some of the things that which can bring about such an exciting thing, action-packed thing, is the nervous system. We have seen how nerves act, how nerves can bring about coordination, how cranial nerves are there, spinal nerves are there, how areas in the brain coordinate these two. And we have seen how some of the actions coupled to these activities is very quick, and the response appears in milliseconds. Response appears in milliseconds. So quick. And in the nervous system, one thing shouldn't be forgotten. The receptor organ and the effector organ are connected. There is a sort of wiring, let us say. Because of the wiring only, you find the nervous system functions in a manner in which it is different from the other system that we are going to discuss now. We will be discussing effects of certain chemicals on the body. Those chemicals are called hormones. Hormones are released by, as you know, I needn't tell you all that, endocrine glands. Endocrine glands found inside the body in various positions. In various locations. They might, I said, affect the functioning of another endocrine gland or they may not. They may have targets on the entire body, all cells of the body, or they may not. They may bring about an increase in the activity of the certain cells, they may not. They may even inhibit. But the time taken for all these things is much more than a millisecond. Somebody would say, I beg to differ from you. If you say that, if someone says that, I will say that is correct. Because responses are very quick in some. We have learned about alarm responses, flight or fight responses created by a couple of hormones released by the adrenal medulla. Not that responses cannot be generated quickly. Most responses generated by hormones are delayed, takes a long time, maybe a week, maybe a month, maybe several years. Maybe the effect will be felt lifelong. Endocrine glands, several are there, but they are located in different places in the body. If you start from the head, you find pituitary comes first. And if you come down to the neck, you find thyroid comes next. Inside the body, find the pancreas comes there, adrenal glands come there, thymus comes there, gonads come there, testis and the ovary. Among these you find some of them are true endocrine glands. What do you mean by that? They secrete only hormones. What else do you expect them to do? Well, there are other glands which carry out a couple of more functions other than those created by endocrine glands. What functions are there? Well, refer to the gland and then to the function. Let us see. If you take the testis in the male, you find not only it produces hormones necessary for growth and development during puberty, it also produces sperms. Sperm production is not an endocrine function. Go to the ovary in the females. Not only the ovary produces the ova, fertilizable structures by the sperm, it also produces female sex hormones called estrogens or progesterone or whatever it is. So hormone producing, non-hormone producing actions are completed by the same organ. This is very important. Now, you say hormones are chemicals. Their action is slower. They have to pass in the blood. They have to reach the target cells. They have to bind to the receptors of the target cells. They have to enter the target cell, bring about a change in the metabolism of the target cell. This is a time-consuming process. It is. And different hormones behave differently. Means different types of hormones are there. Well, based on their chemical nature, you find two types of hormones can be distinguished. One is amino acid derivatives, other is steroids. Amino acid derivatives can be just amino acids themselves. They can be small peptides composed of a few amino acids joined together by peptide bonds. They can be large peptides which we very often distinguished as protein. Most hormones made in the body are this. Most humans' hormones are amino acid derived. Steroid hormones are produced by adrenal gland and gonads. Steroid hormones are made from cholesterol, which is the starting substance for making steroid hormones. So very few steroid hormones are there. Adrenal gland produces a couple of them. Each gonad, the male gonad and the female gonad, produce a couple of them. So those are different when compared to the amino acid derivatives, which outnumber the steroid hormones. Once we know, how can we study the hormones in the laboratory? Well, you require endocrine glands. Is it enough if you have endocrine glands? No, not enough. Endocrine glands in sufficient quantity. Sufficient quantity. If you want 1 kilogram of endocrine glands together, they must come from 8 to 9 persons. 8 to 9 persons. And all the tissue that you collect from these need not be endocrine itself. You have to isolate. You have to isolate the compound, subject to biochemical analysis, and characterize whether it is this or that or that or that. So it is a quite a laborious process. Laborious. We mentioned how many endocrine glands are there and all that. Referred to a couple of them. Some located in the head and some located in the other parts of the body. We shall start our study with an endocrine gland which is called pituitary. Pituitary is a very small gland, of this size once again of the pea. Very often, pituitary gland is described by a physiologist as a pea on a stalk. The stalk here, with the help of which the pituitary hangs down, is called the infundibulum. Infundibulum is a nervous part of the brain, part of the floor of the brain, part of the floor of the diencephalon of the brain. Diencephalon is the forebrain. Hangs down. One part of it is merely attached as an appendix to this one. So you can identify two parts in the pituitary. One is anterior lobe, the other is posterior lobe. Anterior lobe is called adenohypophysis. Adeno, wherever it comes, it refers to a gland. Hypo is below, physis is a process. So glandular process hanging from the brain proper. Posterior lobe is often called neurohypophysis. Some physiologists object to this expression for reasons best known to them. So neurohypophysis, adenohypophysis. Sometimes the adenohypophysis is called pars distalis and this is called pars nervosa. Pars is part. A picture of the pituitary. Pituitary is not just hanging by an infundibulum. Well, it is protected, highly protected. In a bony capsule, of one of the bones of the skull called the sphenoid. Cella turcica, an item for a multiple choice question. Infundibulum, an item for a multiple choice question. Matching the two, 1, 2, 3, 4, the other items can be written out and questions can be made. Well, I shall just represent the pituitary, just follow me. Pituitary lies just behind the optic chiasma. Optic chiasma, this is in a sagittal section, or VS, vertical section. This part is the hypothalamus. This is the pars nervosa. Posterior lobe. I have dotted this portion purposely. This is the pars distalis or adenohypophysis, which is glandular. You mean to say this is not attached to the brain? No, that is not attached to the brain. As I said, it is an appendix attached to the posterior lobe. The posterior lobe. So this is the infundibulum, the stalk by which the entire pituitary hangs, both anterior and posterior together, but the anterior pituitary doesn't have any direct connection. This is the hypothalamus. Here are some of the nerve cells, neurosecretory cells, two groups of them. I have drawn only one, more can be drawn. This is one group, that is the other group. This is called supra-optic nucleus. We have repeatedly mentioned when discussing the structure of the brain that the word nucleus here has a different meaning than found in cell biology. This is supraoptic nucleus, this is paraventricular nucleus. We have shown that every nerve cell is a secretory cell. These are not exceptions. The secretions of a nerve cell, if it has a long axon, under axonal transport it is carried to some other place. We have seen how acetylcholine, a neurotransmitter, is actually produced by the Golgi apparatus of the neuron and is transported by the axonal transport into the synaptic end, synaptic vesicles. Here also. But the secretion is different, that is all. The secretions are peptides, small peptides. One is making one group of peptides, the other is making another group of peptides. So these are, shall we say, hypothalamic hormones or neurohormones. Neurohormones produced by the two groups of nuclei come down and their axons end in telodendria. Telodendria are distributed over a big plexus of capillaries, which forms a part of the hypophyseal portal system. A portal system we have seen in the digestive system is one where veins start in capillaries and end in capillaries. Normally, veins start in capillaries but do not end. In a portal system, they start. Not only they start in capillaries, they end in capillaries. We have seen how such a hepatic portal system collects digested foodstuffs from the stomach and intestine and carries it to the liver. Hepatic portal system. Lower vertebrates also have another system, which is called the renal portal system, absent in humans. Here, it's a hypophyseal portal system because the whole pituitary is named hypophysis. I particularly checked myself for mentioning this word later because hypophyseal portal system ought to be mentioned in connection with the capillary system of the pituitary. What is this made up of? Pars distalis. It is made up of secretory cells. Not nerve cells, no. How many types? Well, there are several types. You can call them alpha. Generally, the distinction is by making use of a Greek alphabet, alpha cells, beta cells, and then, or you can call them acidophils, taking acidic stain, basophils, taking a basic stain, or neutrophils, not taking any stain. Anyway, a section of the pituitary can be examined under the microscope, high power, in the college laboratory, and you can identify different types of cells there if the pituitary has been stained in multiple stains. Mallory's triple. Not at all difficult. I have been doing that in the laboratory. Only thing is the section must be good, that is all. But this is not composed of nerve cells, no. It is not connected to the brain, no. It is having a different structure. Oh, this has one structure and that has another structure. Yes. If they are structurally different, we guess they must be different in origin also. Well said. Right you are? They are different in origin. Just see what happens during embryonic development, not in the adult. This is the head. This is the buccal cavity. This is the developing brain. The developing brain produces a downward growth. This is the buccal roof. It produces an upward growth. This is the buccal cavity. This is the lower jaw. Now, this structure is the Rathke's pouch. Rathke's pouch is the one which dissociates itself after some time, continues its attachment to the posterior lobe of the pituitary and develops into the anterior lobe of the pituitary. So, this is nervous in origin, this is non-nervous, epithelial in origin, epithelial. So, the origin and the structure, naturally, the structures are different, you find. They might be different in origin, but they will be certainly different in function. The functional differences between the two will be discussed presently. Different types of cells are there, as I said, in the anterior pituitary, and they can be identified. Their products cannot be identified by microscopy. It requires a different technique. Several hormones are released by them. I said most of them come under one category, some others come under other category. It was not a reference to pituitary alone. All pituitary hormones are amino acid derivatives. They are either small peptides or large peptides, proteins, with a certain number of amino acids joined together to form a peptide or protein. Several hormones are there. How many are there? Well, 6 to 7. Why 6 to 7? Let us find out what is the problem with the seventh hormone. 1. Growth hormone. Somatotropic hormone. Somatotropin. TSH. Thyroid stimulating hormone. Thyrotropin. ACTH. Adrenocorticotropin tropic hormone. Adrenocorticotropic hormone, ACTH, adrenocorticotropin, follicle stimulating hormone, FSH, LH, luteinizing. Careful about the spelling. Luteinizing hormone. LTH, lactotrophic hormone. Somebody will ask me a question now. If you don't ask, I will ask myself and answer. There are two ways of writing this. Trophic. You can write TROPHIC or TROPIC. This trophic would mean nutrition. As you have heard about autotrophs and heterotrophs, chemotrophs, etc. It may indirectly mean that tropic is stimulating. Many textbooks use this spelling and a few textbooks use this spelling. I think I prefer this because those are the stimulating factors. Trophic and tropic. Well, you said some problem with the seventh one. MSH, melanocyte stimulating hormone. Of these you find follicle stimulating hormone, and many authors include this also, lactotrophic hormone. These are often called another word, tropic hormones. Gonadotropins, let us say. Make it single. 2, 3, 5 to 6 are all simply tropic hormones. What is that? That is also tropic, this is also tropic. Should you once again take up two terms to identify them? Well, it is necessary. Gonadotropic hormones are those which stimulate the gonads. Gonad stimulating hormone. These hormones stimulate any endocrine gland. So, among these you find growth hormone is the only hormone that is not tropic in its effects. All the others are. Melanocyte hormone is a problematical one because it is said to be secreted by intermediate lobe. Well, we didn't refer to it, we will refer to it. Intermediate lobe of the pituitary, which looks vestigial in humans. So, many textbooks don't mention this at all, or they just give a lateral explanation for that. So, let us take up the first hormone, which is a non-tropic hormone released by the anterior pituitary. Growth hormone, somatotropic hormone. Soma is the body. You have heard about somatic and germinal cells. Growth hormone concerned with body growth. So it's target cells, all cells of the body. Targets, which means have receptors, have membrane receptors. Growth hormone, well, responsible for entire growth, growth of reproductive organs, growth of features which identify maleness and femaleness. In general, you find all aspects of growth. Now, any hormone, whether it is growth hormone or something else, can have two effects. One is that the hormonal concentration may go down due to several reasons. Another is the hormonal concentration may go up, rise. Each hormone has a definite value. There is a borderline at which it must exit to create an effect. Threshold level. If the hormone is not to be found in a threshold level, it is under secretion. If the hormone is to be found above the threshold level, it is over secretion. Under secretion is called hyposecretion. Hyposecretion. We shall use this terminology for all the forthcoming hormones also. Hyper, additional, extra, excess, hypo, lower, inadequate, not enough, less than the normal quantity. Supposing a hormone is found below its normal threshold level, could it create problems? Supposing a hormone is found in excess levels, could it create problems? Yes. Problems are created by both, and names are associated with each of these problems. You find for hyposecretion there are, for hypersecretion there are. And one more cog in the wheel here is that when is the secretion less? When is the secretion more? Is it before attaining childhood, before puberty, or after puberty? Hypersecretion, is it before puberty or after puberty? So in humans you find when such a study is made, well, this whole study is endocrinology. This is with specific reference to hormonal effects.